Hello again, and welcome back to the January 2009 BY1 paper. We're on to question number 3 now. The first part here just tells you about the diagrams in the question. Okay, so the examiner is telling you there is a molecule of starch and a molecule of cellulose. Okay, starch and cellulose. And the structures are shown underneath that first statement. What I want to help you out with to start with is just to go through these structures shown in the question. Okay, now I've given you structures of starch and cellulose in the notes. Okay, so if I just pull the notes up, okay, that are on the app. Okay, this is the diagram I've put in of cellulose, okay, to start with. All right, now the diagram here, I've decided to use detailed images of the beta-glucose monomer that makes up cellulose. Okay, but if I flip back now to the exam question, you can see that the examiner has actually used an abbreviated version of the beta-glucose monomer, and indeed that of the alpha-glucose monomer, which is found in starch. Okay, so you do have to be aware that there are a number of ways of representing molecules and their structures. Okay, so just be familiar with the different ways that your teachers and examiners can represent these structures. Okay, right, if we scroll down to the actual questions now, okay, we've got part A, and it's asking you to name monosaccharide X and its form. So monosaccharide X there is in the cellulose structure. Okay, now I've already told you that cellulose is made up of beta-glucose. Okay, so I've actually given you the answer to part A already, but I just want to explain really what is meant by form. Lots of people get a little bit confused as to the word used in this question. Okay, all it means here is isomer. Okay, so the answer to part A, okay, would be beta glucose. If I just type that in for you, there we go. Okay, so there's the answer done. If we move on now to part B, it's asking you to name the bond formed between the two hexose sugars. Now, I just want to underline their hexose sugar, okay, and explain exactly what that means. Hexose sugar is a term I've talked about in the notes that I've produced. Remember that there are many ways to classify sugars. One way is to classify them according to the number of carbon atoms they have. Okay, and that's what's going on here. A hexose sugar is a sugar that actually has 6 carbons in its structure. Okay, so basically, if we look at the structures of starch and cellulose, as you know, starch is made up of alpha glucose, cellulose is made up of beta glucose. Both alpha and beta glucose are hexose sugars because they have 6 carbons. And if I just arrow the carbon number 6 there in the alpha glucose monomer, and the carbon number 6 is in the same position in the beta glucose monomer of cellulose. Okay, just to help you out again, that point there represents carbon number 1, okay, on the alpha glucose, and the carbon number 1 is in the same position there on the beta glucose monomer. Okay, so again, you've got to be comfortable with the different terms used to describe sugars. Okay, Okay, so that's hexose sugar explained. So what's the name of the bond formed between the two hexose sugars? Well, let me arrow them up for you. There is a bond, there's another one, and there's another one again. Okay, and the same for the alpha glucose. There's the bonds between the hexose sugars, or the glucose monomers in this case. The bond, okay, is the glycosidic bond. Okay, and if I just type that in for you. Okay, there it is, glycosidic bond. There are a number of bonds that you need to remember for your A-level biology, and glycosidic, of course, is just the name of the bond that forms between sugar or glucose monomers in carbohydrates. Okay then, if we move on to part C, now the command word in this question is state. Now you do need to view my video tutorial on command words. Okay, so I won't go into the exact meaning of state in this video. If you just check out the video on command words, and that will be explained in there for you. Okay, so part C then, you need to state two structural differences between starch and cellulose. Now there are many structural differences between starch and cellulose, okay, but there's a little trick to answering this type of question, okay. Let me give you an example of a structural difference between starch and cellulose. Okay, starch is made up of alpha glucose and cellulose is made up of beta glucose. Okay, now what the examiner wants in this type of question is that he wants you to state concisely the differences between one feature of starch and cellulose together. Okay, so let me write in the structural differences I just mentioned as the examiner would want you to do it. Okay, so there's the answer in. If I read it out to you, cellulose is made up of beta glucose and starch is made up of alpha glucose. Now that answer states the two differences together. That's what the examiner would want to see. Okay, he wouldn't want to see, for example, cellulose is made up of beta glucose. If you just put that in, you would not have achieved 1 mark. Okay, so the examiner does want two structural differences. Okay, so I'll type in another structural difference and we'll have a little chat about that. Okay, there's my second structural difference. I've said cellulose has 1,4 glycosidic bonds, while starch has 1,4 and 1,6 glycosidic bonds. Okay, now if I pull up the notes that are in the app, okay, I've got a diagram here of a mylopectin. You've got to remember that starch is made up of two different polysaccharides. One is a mylopectin, and the other one, if I scroll up, is amylose. Okay, now in amylose you have just the 1,4 glycosidic bonds. Now there's the 1,4 glycosidic bond there. Okay, but in a mylopectin, what you have is your 1,4 glycosidic bond there, shown by the red arrow. But you also have this 1,6 glycosidic bond there. Now remember, wherever you get a 1,6 glycosidic bond, that represents a branching point within the structure. Okay, so that's a very obvious structural difference between starch and cellulose. If I just scroll down to the cellulose structure, okay, there's a diagram of cellulose, okay, and what you have is just the 1,4 glycosidic bonds present. There is no branching in cellulose, okay, so you do not get a 1,6 glycosidic bond. Okay. Right. Okay. If we go back to the question now, we've answered part C. We've got our 2 marks. But what I want to emphasize at the moment is there are a lot of other structural differences that we could have quoted or stated in this question. When we have a look at a mark scheme at the end, I'll show you the number of different structural features that the examiner would have accepted. But let's finish off the question. We're coming on to part D. This is the last part. And it's stated now, starch is a compact storage polysaccharide. Cellulose has a structural role in plant cell walls. Hopefully you know that. Okay, starch is a storage polysaccharide. It's a storage of energy, of course, in the form of glucose, whereas cellulose has a purely structural role. Okay. So now you're asked to describe, and of course that's another command word there. Describe how cellulose units. Okay. Now the examiner's being careful here. He's called it cellulose units. He couldn't put beta glucose here because he would have actually then given you the answer to part A. All right. So the examiner's being very careful not to help you out here. Which is right, I suppose. Okay, so the cellulose unit, of course, is the beta-glucose monomers. Okay, so you're asked to describe then how cellulose units are arranged in a complete molecule and how this achieves, or how this arrangement gives cellulose a high tensile strength. Okay, the major thing that gives cellulose its high tensile strength is the large number of hydrogen bonds that hold the structure together. So if I pull up the notes again, this is a simplified diagram really of the structure of cellulose. So what you've got there with the green line, that represents your long straight chain of beta-glucose monomers. Okay, and each chain of beta-glucose monomers is held together by hydrogen bonds. Okay, so the hydrogen bond is being formed between adjacent or neighboring chains of beta-glucose. Okay, now those hydrogen bonds are being formed between the OH groups. Okay, so there's an OH group there, an OH group. Okay, just to remind you that in the structure of cellulose you have every other beta-glucose monomer being flipped by 180 degrees, yeah, and that causes the OHs, or the OH groups, to point down and to point up. Okay, so that's why I've put the red arrows on these notes to show you the alternating up and down arrangement of the OH groups. Now those OH groups then allow these hydrogen bonds to form between the neighbouring chains. Okay, so that is one marking point really within this question, is to state that there are long chains of beta-glucose that are linked or joined by hydrogen bonds. Okay, so if we go back to the question, okay, and we'll just type that answer in. Okay, so we've entered one correct answer there. Cellulose has long chains of beta-glucose monomers linked by hydrogen bonds. Okay, so some other arrangements within glucose. We've mentioned some of them previously. You would have allowed a mark for adjacent glucose monomers or molecules are rotated by 180 degrees, as I previously mentioned. So we can actually type that on. Okay, so there's our second answer. Glucose molecules are rotated by 180 degrees. Yeah. You could have put in that question, of course, or in your answer, the term microfibril. Okay, I've mentioned microfibril there. Just remember that a microfibril is formed when you get many long straight chains of beta glucose being hydrogen bonded together. Okay, so they would have accepted the formation of microfibrils as well. Okay, so in our answer then, the answer here, cellulose has long straight chains of beta-glucose monomers linked by hydrogen bonds. That answers the high tensile strength part of the question. It's the hydrogen bonds that give the high tensile strength in cellulose. And the second answer there is asking about or answering how cellulose units are arranged. Okay, we've said that there are glucose molecules are rotated by 180 degrees. Okay, right, so that's the end of question 3. If we have a quick look at the mark scheme, okay, so there it is. Part number 1 there, or part A, beta glucose. The next one is glycosidic. Now, part C, as I told you earlier, there were a lot of options that the examiner would allow for the structural differences between starch and cellulose. There they are. You can read them yourselves. Please try and commit those to memory. Okay, it's quite an important set of notes there really for you to make. Just to highlight to you, right at the bottom of the mark scheme, the examiner has said there must be a direct comparison. Okay, if you didn't make a comparison, you wouldn't have got the marks. Okay, right, on to part D then. Straightforward enough. We've mentioned the long straight chains and hydrogen bonds. We've talked about the glucose molecules being rotated by 180 degrees. Okay, we could have put in, as I mentioned, microfibrils. Okay, and there was also one other option there where you could have stated where the hydrogen bonds form between, and they were between the hydroxyl groups of adjacent chains. Okay, right, okay, that ends question 3. I hope you found it of benefit.